Welcome to your module on cellular regulation. We're going to start off talking about cancer and the different processes involved pathophysiologically as well as treatments. And then there's future presentations which will focus more on the assessment and nursing care of these patients. I've spread it out over 4 different presentations so that it's not so intense when you sit down. You can do it in chunks instead of one like mega long session about it. So hopefully that will benefit you all and you will be able to learn better that way. Learning outcomes for this lesson, and that includes the future presentations pertaining to cancer, are listed here, and I've gotten those from your textbook this time instead of your syllabus so that you'll have a more targeted area of study. So look through these, make sure that you are familiar with these various topics. I'm not going to sit here and read them to you. Y'all are very capable of doing that, but these are going to be the areas that you should focus your studies on. So let's begin with talking about a patient who happens to be a friend of yours, actually. She's trying to get back in nursing school. As a side note, she comes to you because she knows you already are in nursing school and you are very familiar with um, the warning signs of cancer, which you'll learn about in a little while. But she wants to know about this spot that she has on her back. She's worried. She knows her mom had a history and died of cancer, and she wants you to tell her what it is that you know about cancer. So take a few minutes, and I want y'all just to jot down on a piece of paper 5 things that come to mind when you think about cancer. So pathophysiologically, we know that cancer is a group of diseases that are characterized um, by an uncontrolled and unregulated growth of cells. We know that cancer does not discriminate. It affects both sexes. It affects races of every sort. It affects young, old. It does not have a favorite. It's the second most common cause of death in the United States, and that's following heart disease. But fortunately, the survival rates for cancer are much greater than they have been in the past because of research, and science has advanced as far as treatments go. Some cancers, though, like skin cancer, are still on the rise, and that's a cancer that prevention plays a large role in because we can provide education on how to prevent skin cancer, things, lifestyle changes that people can make to reduce that risk. So even though we have all these great resources, scientifically speaking, sometimes just the simple part of education can be the largest influence on whether or not patients develop cancer. The incidences of many cancers have declined, again, largely as a result of these preventative efforts. Um, Other types of cancers, such as kidney or thyroid cancer, non-Hodgkin's lymphoma, unfortunately, those types of cancers have been increasing. We know that more than 14.5 million Americans are alive today who have a history of cancer, and they're either disease-free, they're in remission, or they may be currently under treatment but with a very favorable prognosis. Regardless of what statistics can tell us about the survival rate of cancer and which cancers are on the decline and which ones are still on the rise, statistics can't adequately give us a description about the total effect of finding out you have cancer and what that means to patients and their caregivers and their families. We're going to dig into a little bit of the biology of cancer. I don't want this to overwhelm you, but I do want you to have an understanding of what occurs at the cellular level that leads to the development of cancer. If you can understand it and try not to get overwhelmed with it, then the different treatments will make a whole lot more sense, and the different side effects of those treatments will make a whole lot more sense. So again, don't get overwhelmed. Just try to understand the basic processes of how cancer develops. We know that this starts with two different major dysfunctions, which include a defective cell proliferation stage or a defective cell differentiation stage. Cancer wreaks havoc on the ways that cells typically function. Normal body cells have a very predictable pattern of growth, division, and death. It occurs in a very orderly fashion because our bodies are trying to maintain an equilibrium or a homeostasis of sorts, and so our bodies have a predictable pattern of cell growth, replication, and death. But with cancer cells, they have a mind of their own, and they don't follow this predictable pattern, and they end up multiplying, forming new blood vessels that kind of feed that cancer and continue its growth. Normally, most tissues are going to contain a population of undifferentiated cells known as stem cells that eventually do differentiate and become mature functioning cells of that specific tissue. So a lot of times you'll hear about stem cell treatments with cancer, or you'll hear about harvesting stem cells from cord blood and that type of thing. Those stem cells are undifferentiated cells. Eventually they will differentiate and they'll target that specific tissue that they were retrieved from or that they're located in. But that's just a little bit of FYI about stem cells for you. There's a link here that I need you to watch. I'm not going to try to play it in the presentation because I know it's not going to work, but make sure you go back and view this because it's going to help explain the difference between normal and abnormal cells. I want to talk a minute about what cellular proliferation actually is so that you can understand when a defect occurs, how that impacts the body. So cellular proliferation, it includes factors like nutrients, the temperature levels, pH, your oxygenation. All of those factors contribute to various mechanisms that control cellular proliferation. And cellular proliferation is just an increase in the number of cells because of cell growth and cell division. Okay, so those mechanisms that are controlling that are what determine whether proliferation occurs and the rate of proliferation, so on and so forth. But with cancer, there's a defect in that cellular proliferation. And so, whereas in normal cells can respect their boundaries. They are not going to go invading territories that aren't their own. They're going to stay where they're meant to stay. So if it's, for instance, cells that are within your liver, they're going to stay where they're supposed to stay. They're not going to migrate. That's thought to be as a result of boundaries that are established from neighboring cells. But with cancer cells, they don't care. They don't have any regard for boundaries. They go where they want to go. They grow how they want to grow. They have no rules. So it kind of made me think about having a nosy neighbor, right? So these cancer cells are nosy. They want to go other places. They want to find out what's going on around them. They want to see if they can be bigger and better than those average cells that live next door. And this is all a result of genetic mutations. Genetic mutations that are a result of either an inherited um, mutation or a mutation that's caused by something like a virus, hormone, lifestyle, um, anything else that can impact abnormal cell growth. Again, typically when we have cellular differentiation, and that's when those cells are dividing, right? It's an orderly process. Cells are maturing from an immature state, and they're differentiating into mature cells. And under normal conditions, that's going to be the process. They're not going to revert back to a previous state. But with cellular differentiation that has a defect, they can revert in between the different stages. Cancer involves the malfunction of genes that control how cells differentiate and proliferate. Um, there's two types of genes that can be affected if there's a mutation, and those are the proto-oncogenes and tumor suppressor genes. Those are the two genes that are targeted when a mutation occurs. Normally, proto-oncogenes act to regulate cellular processes so they can help promote growth or whatever it is the process that's meant to occur. That proto-oncogene is there to help regulate that process. Tumor suppressor genes exist to help do exactly what they say. They help suppress growth, and so those are their normal functions. But when there's a defect, as a result of some type of mutation, then these genes start acting crazy. Okay. Typically, again, with that proto-oncogene, they have a built-in genetic lock that helps them to function normally. But when that gets mutated, instead of being a proto-oncogene, they turn into something called an oncogene. That lock that existed becomes unlocked, whether it's through exposure to a carcinogen or some type of virus. That mutation occurs, and those abilities and properties that the cell had in that proto-oncogene state change. Now, with tumor suppressor genes, they normally exist to regulate cell growth, but that mutation that targets that gene makes it inactive, and that causes a loss of the ability to suppress tumor growth. In this slide, you can just see the initiation, promotion, and progression phase that we've been discussing. A lot of times we think that cancer is just a random, haphazard event that occurs very quickly, but in a lot of cases, it's actually occurred over a very long period of time, as these mutations have affected the genes and normal processes become abnormal. So in that initiation stage, or that very first stage of cell development, when we're speaking about cancer, this is the stage that the mutation is going to occur at the cellular level. There's some type of change that has occurred in that gene that's led to that, and that's called initiation. So that cancer is being initiated. Those cancer cells are arising from those normal cells as a result of the changes that have occurred in that gene. Sometimes these gene mutations are inherited. Most of the time they're acquired. And when they're acquired, they're as a result of a carcinogen. Carcinogens are those cancer-causing agents that can produce changes in the cell. They can make the cell behave differently than it's supposed to behave. They can be chemical carcinogens, radiation, viral carcinogens. Some chemicals that have been identified as carcinogens over the past have included formaldehyde, arsenic, benzene. It's interesting that the first chemicals that were identified to cause cancer were discovered like in the latter part of the 18th century. It was discovered that chimney sweeps had a higher incidence of cancer of the scrotum associated with their exposure to soot. And that's how that relationship first was established. People that are exposed to certain chemicals over a consistent period of time, so repeated exposure, they tend to have a greater incidence of certain cancers than others. And because there is a long phase from having the cancer initiated until it is able to be detected, sometimes that makes it difficult to identify what exact carcinogen is responsible for that cancer developing. Oddly enough, radiation, which is something that we use as a treatment for cancer, can in fact cause cancer in almost any human tissue because of how it damages the DNA. Certain types of cancer malignancies that are associated with radiation are leukemia, lymphomas, thyroid cancers. Back when the atomic bomb happened in Hiroshima and Nagasaki, that resulted in a higher incidence of cancers from that exposure to radiation. Occupations like radiologist or radiation chemist, uranium miners, they're all at a higher risk for cancer because there's been evidence that there was more cases of bone cancer in those occupations. Viral carcinogens, think about like HPV. How much do you see on TV about HPV virus and that there's a vaccine or now to help prevent the development of HPV? And some people have questions about it because of their personal beliefs regarding the vaccine, but it exists to help prevent cancer because we know that HPV is a viral carcinogen. Hepatitis B, the same situation, except it targets the liver. HIV is associated with Kaposi sarcoma, and Epstein-Barr virus is associated with Burkitt's lymphoma. So there's certain types of viruses that we refer to as oncogenic. Remember, that's what happened after that mutation in the gene affected that proto-oncogene. It made it oncogenic. So these are oncogenic viruses, and we know that they can transform cells that they've infected and that they can cause malignancy to occur. Your patient or friend, however you want to refer to it in this situation, she has expressed that she's scared. She knows she's been spending a lot of time in the sun. She hasn't been using sunscreen, even though she promised her dying mother she would. And she is asking you, what do you know about how the sun causes cancer to develop? So take a few minutes and think about what you've been told in your life about the exposure to UV rays and sun and how it can impact you developing skin cancer, or if you know somebody that has had skin cancer as a result of their exposure. Just take a couple minutes to think about that. Hopefully you wrote down that you know that UV radiation is a carcinogen. It's a known carcinogen. And carcinogens can be chemical, they can be radiation or viral, as we talked about. But we know that UV radiation, especially, has been long associated with melanoma or the malignant skin cancer, as well as other types of skin cancers. We really have seen an increase in melanoma, and that's very concerning. Um, we we know that it's probably more than just the UV rays that cause it, but we know that prevention is key in reducing the cases of melanoma, and one of the easiest things you can do is to avoid excess sunlight exposure or excess artificial UV exposure, such as in tanning beds. Moving on to the promotion stage of the development of cancer. We've talked about the initiation stage and how the genetic mutations target certain genes. Now we're going to talk about the promotion phase, and an important distinction between the initiation and the promotion phase is that in the promotion phase, that is considered reversible, right? So we may not can control when these changes are initiated, but we can help control how they are promoted. So that's that key concept that occurs when we talk about cancer prevention and how cancer prevention influences the rates of cancer that we see. So about a third of deaths related to cancer in the United States are related to tobacco use, an unhealthy diet, physical inactivity, and obesity. We have certain kinds of carcinogens that are capable of initiating cancers, and we have others that are capable of promoting them, and then some can do both. Cigarette smoke is an example of what we call a complete carcinogen, which is a carcinogen that can both initiate and promote cancer. The latent period of cancer can take anywhere from 1 to 40 years before it can develop to the point that it can be detected. Right now, the smallest tumor that can be detected by a diagnostic measure like an MRI, for instance, is about a half a centimeter. A tumor that is 1 centimeter, which is what we can detect typically by palpation, contains 1 billion cancer cells. So by the time that it gets to the point that we can palpate it, it's already containing 1 billion cancer cells. That's kind of scary, isn't it? The progression phase of cancer is when we talk about the growth rate of the tumor. It's increasing, the invasiveness of it, and the metastasis of it. It's that final stage in the natural history of cancer. It's that metastasis is that phase where, that stage where it's spreading to a distant site. And usually that's going to occur, you're going to know that the metastasis has occurred because the patient has some new onset of, whether it be like a bone pain indicating a metastasis to the bone, or a change in their bowel or bladder tone indicating metastasis to their nervous system. Main sites of metastasis are shown in the slide: brain, cerebral spinal fluid, lung, liver, adrenals, bones. Those are those areas that are most frequently found to be areas of metastasis. In the progression phase, that metastasis process is going to begin with that tumor rapidly growing. That it's just growing, growing, growing. It's getting its own blood supply. It's detaching and going around and invading other areas and tissues. And those cells that have detached, like I said, they're going and they're invading other areas. They're nosy neighbors who are going around the neighborhood, going and checking on everybody else. And most of the time, the cells that are able to do this don't survive, but occasionally, the ones that do survive and are able to create an environment where they can continue to grow and develop, those are the ones that are going to cause the most problems. You've heard of the phrase sentinel node. So a sentinel lymph node is that first node that the tumor cells approach as they go throughout the lymphatic system. A biopsy of that sentinel lymph node can help in determining how widespread the cancer is. Tumor cells can get trapped in that first lymph node, and that biopsy that's done, again, can help determine that extent of the cancer. The procedure is actually less extensive, requiring the surgeon to remove fewer lymph nodes. And then the tumor cells themselves, sometimes instead of going to that first lymph node, can actually do something called skip metastasis, which is when the tumor cell travels to lymph nodes further distant. So some malignancies like esophageal cancer, for instance, that's what occurs in that case of metastasis. It's a skip metastasis. So it bypasses that initial lymph node and travels to other areas, to other nodes. Humans have these fascinating immune systems that help detect invaders, whether it be viruses, bacteria, help respond to cancer cells. And so the immune system, it's supposed to respond to these cells. It's supposed to reject and destroy these cancer cells. But we know that in many cases it's not adequate as these cancer cells continue to develop. And what occurs is that these cancer cells, they have changes in the antigens that are on their surface. So they're called tumor-associated antigens. You can see in the slide how the normal cell has your nice round normal surface antigens, but the cancer cells have these spiky ones that are called tumor-associated antigens. And it's the immune system's job to respond to those. But again, sometimes it does a good job and sometimes it doesn't. The immune system acts to respond to these tumor-associated antigens. They're supposed to detect and destroy the cells that are developing, and under most circumstances, that's going to occur. But just like anything, there's always an exception, and in some cases, there are these mechanisms that make it possible for cancer cells to evade the immune system altogether. Whether it's through a suppression of factors that are responsible for stimulating T cells, or whether it's because there's weak surface antigens that cancer cells can sneak by. One of these things are going to allow for the cancer cells to escape the normal immune response. Oncofetal antigens are substances which are produced by tumors, and also they are produced by fetal tissues, but at a much lower concentration than adult tissues. Certain oncofetal antigens can serve as tumor markers. Examples of oncofetal antigens include CA125, which can help serve as a tumor marker for ovarian cancer. PSA for prostate cancer is another one. There's also various ones for breast cancer and colon cancer, as well as pancreatic and gallbladder cancer that you can see here listed. They're not, oncofetal antigens are not 100% specific for tumor reoccurrence, but they are useful in many cases to monitor, the effects of therapy and determining sometimes whether or not that tumor has reoccurred. So the patient wants you to tell her, or the friend wants you to tell her if her cancer is the bad kind or not. So think about what you know as far as the differences between benign tumors, and malignant cancer tumors, and how we can detect the difference. We divide these types of tumors that are detected into categories of benign or malignant. And the difference largely being that malignant tumors have that capability to invade and they can spread and metastasize. That is the major difference between the two types. When we refer to tumors, we classify them, and we can classify them by their an anatomic site. We can classify them based on histology, and we can classify them based on the extent of the disease. Classifying the tumors in this manner helps to highlight the risk of that cancer and what the impact of that cancer is going to be. It also helps for communicating between different healthcare teams and in establishing a treatment plan, and again determining the prognosis, and also in comparing groups in a statistical manner. When classifying a tumor based on its anatomic site, you're going to be identifying that tumor by the tissue of origin. Where did this tumor originate from? Carcinomas can originate from the skin or glands, which is considered the embryonal ectoderm. They can arise from mucous membranes of the respiratory tract or the GI and GU tract, which would be endoderm. Sarcomas are originated from embryonal mesoderm, which is the connective tissue, muscle, bone, and fat. And y'all learned about ectoderm, endoderm, mesoderms all in pathophysiology, hopefully, or Bio 168, 169. And we also know that there are lymphomas and leukemias that originate from the hematopoietic system. So when we talk about the different anatomic sites, that's what it's referring to. In reference to the histologic classification or grading of tumors, that's when the pathologist is looking at the appearance of the cells, the degree that the cells are able to differentiate. How closely do those cells resemble what the normal tissue of origin looks like? If a tumor is very poorly differentiated, then we know that's going to have a poor prognosis than one that looks more like the normal cells do. And there's 4 grades when we refer to this histological staging or grading of the cell, and it's grades 1 through 4, which vary from the cells very slightly are different than normal cells, and they do have some appropriate differentiation, to grade 4 where their cells that are immature, they're not differentiated, and you can't determine what the origin of that cell is. In regards to clinical staging, which is what I really want y'all to be familiar with, is 0, which is cancer in situ, up through 4, which is metastasis. And this is the amount that the cancer has metastasized. So is it contained and it's not spread anywhere, which would be 0, or is it limited just to that tissue that it originated in, which would be a 1? Has it spread a little bit in a limited area, which would be a 2, or has it spread extensively, locally, and regionally, which is a 3, or has it progressed to other areas and tissues so that it has metastasized and is considered a 4? You need to know this classification of cancer as far as the TNM classification system. I mentioned it at the beginning of the lecture. With the TNM, you have the T for tumor size and invasiveness, the N for spread to lymph nodes, and the M for metastasis. We can't use this for all kinds of malignancies because clearly leukemias are not solid tumors, and so that wouldn't be an appropriate type of malignancy to use this classification system. But if it's a solid tumor, we can use this to classify. So the staging is done either clinically or surgically. They try to avoid surgical staging now unless it's just a type of malignancy that they can't get to otherwise. Clinical staging can be done through different types of scans, ultrasounds, CTs, PET scans. Whereas the surgical staging, of course, requires the actual surgery, whether it be exploratory or a lymph node sampling. But the exploratory phase of staging is being used less frequently. The lymph node staging is going to be pretty prevalent, but that exploratory where they're just going around looking, they don't really have a specific focus. They're just exploring the whole cavity to see what they can find about the malignancy. That's being less frequently used. So once the disease, the extent of it is determined, then you don't change that stage classification. The original description of the extent of the tumor is part of the patient's record, but if additional treatments are needed or if the treatment that's in progress fails and they need to restage that particular tumor or malignancy, then they can do so, and it's referred to as a retreatment staging. And changing gears a little bit now that we've discussed diagnosing the cancer, let's talk about the psychological aspect of finding out that the patient does in fact have cancer. Just the possibility alone elicits a lot of fear and anxiety. Patients are having to undergo multiple tests. They are thinking about if these results are positive, how it's going to impact me, my life and my job, my finances, my family. Am I going to die? All of these things are going through their heads. So even in that pre-diagnosis phase, you really need to focus on listening to what your patient's concerns are, addressing your own discomfort, acknowledging what you feel comfortable with in talking about this with your patient. Do you have baggage? Has cancer impacted you personally? And so as a result, you are coming into that patient-nurse relationship with some of your own fear and anxiety. You need to make sure that you're giving clear explanations about why these patients are having certain procedures. What is this procedure intended to do? What is it going to tell us? Providing written information, because they're going to need reinforcement. They're not going to remember what all you're telling them because they're just in a fog at that time. And if there's an availability of an oncology team that you can refer or have the doctor refer these patients to, that's very helpful. For many patients, the fear of the unknown is actually more stressful than when they actually find out they have the diagnosis. So in managing your own discomfort, you want to make sure that you avoid types of communication that can be closed communication or communication that is going to hinder that patient from exploring how they're feeling. You want to encourage them to talk about how they're feeling if that is something that they're leading you to believe that they're trying to do. So avoid using overly technical language. Use more like layman's terms. Avoid providing false reassurances. You don't want to tell them, oh, I know you're having this test because you think you may have skin cancer, but it's probably nothing. You don't need to worry. That's not helpful. Nor is redirecting them and telling them, oh, we'll just talk about that later, or let's wait until we find out for sure. Let's not worry about that right now. That's closing down the communication. That's shutting down. It's not giving acknowledgement to how that patient is feeling at that right moment. As well as you want to avoid generalizing. Well, you know what? I know that you're upset and I know you're stressed, but everybody who goes through this faces that. That's not helpful either. So what are some things that you can do if you are uncomfortable discussing the patient's fears and their concerns, or you don't know how to talk about it with them? You need to really take a minute, and I want you to think about your own fears and concerns in regards to cancer and death. Think about that for a minute, and also think about potential resources that you could refer these patients to. When talking about establishing a plan of care for these patients, we want to, as far as a nursing perspective, go ahead and initiate a plan of care as soon as we suspect that, as soon as the patient presents and thinks that they have cancer, okay? That's when you want to go ahead and initiate that plan of care. Once the diagnosis, medically speaking, actually occurs, then we can always regroup and readdress whatever needs the patient may have at that time. When looking at patients and determining what needs they have, look at what their potential risk factors are, or what their actual risk factors are. Are they an older adult? An older male patient may be more predisposed, say, to pancreatic cancer or prostate cancer. Is it a female patient? Obviously, they're going to be more at risk for breast cancer. Is it a patient who is immunocompromised or has a history of multiple sunburns over their life? They're going to be more at risk for skin cancer. So think about what risk factors these patients have, and that can help kind of streamline what teaching you need to do and what interventions need to be put into place. Diagnostic studies are going to be selected based on the specific site of where we suspect the cancer. For instance, cervical cancer, of course, pap smears, oh Lord, I want to say colonoscopy, but that's not it. Colposcopy. Thank you. I used to work in a doctor's office, and so if a patient had an abnormal pap smear, then the next step would be to do a colposcopy, where the doctor could do that procedure and look closer at the cells of the cancer. And then if needed, they would progress to what's called a punch biopsy, where they would actually take a sampling of the cervical tissue and send that off for the pathologist to look at and determine if cancer was present. So it's just really going to depend on the site of the cancer. This slide shows you a PET scan, and in the picture, the A is before treatment, the B is after treatment. The more radioactive material that's accumulated shows you where there was a higher level of cancer activity. So clearly the treatment is working for this patient. Again, it just repeats that, diagnostic studies are really going to depend on the site of the cancer. When biopsies are performed, that is when there's going to be a histological examination. The pathologist is going to look at the tissue. Tissues can be obtained various ways, maybe through a needle aspiration or incisional procedure or an excisional procedure. Those are different routes of obtaining those samples. And during that pathology evaluation, that is actually the only definitive means to diagnose cancer, and they can determine whether the cancer is benign or malignant based on that evaluation. So in this situation, the friend tells you that she wants to be healthier. You need to talk to her about different lifestyle changes she can adopt to help prevent cancer or lead to early detection. You would talk about things like reducing and avoiding exposure to known carcinogens, such as cigarette smoke and sun exposure, the importance of a healthy diet, exercise, reducing intakes of carcinogenic foods, such as foods high in nitrites like hot dogs, salt smoked foods, those types of things. Prevention is really the key when we talk about reducing the cases of cancer. We really need to emphasize health promotion and prevention with our patients and identify those potential patients who have risk factors and focus on educating them on things they can do to reduce those risks. Lifestyle habits to reduce risk, they really need to know what cancer screenings are appropriate for that population. If there's a self-examination that is applicable to the patient, they need to be performing that. Knowing the 7 warning signs of cancer and knowing when to seek care if they suspect that they do in fact have cancer. And those 7 warning signs are if a patient notices a change in bowel or bladder habits, if they have a sore that doesn't heal, if they have unusual bleeding or discharge from any body orifice, thickening or a lump in the breast or elsewhere, indigestion or difficulty in swallowing, or obvious change in a wart or mole, or a nagging cough or hoarseness. Those are considered the 7 warning signs of cancer. These just are going through your various lifestyle habits to reduce the risks of cancer.